today we'll be learning about diagram of nephron. Okay, nephron, it includes glomerulus and renal tubule. There are about 1 million nephrons. Coming to glomerulus, consists of tuft of capillaries which gets invaginated into the dilated blind end of the tubule. This is called as Bowman's capsule. So how are these capillaries formed? From an efferent arteriole and afferent arteriole form this tuft of capillaries. So from this capillaries we'll get a filtrate which passes into this Bowman space, from this into the renal tubule, from there into the renal pelvis. Finally the urine passes to the ureter into the urinary bladder. Coming to the proximal convoluted tubule. The proximal convoluted tubule, it consists of a single layer of cells which are attached together by tight junctions. There is extracellular space which invaginates between the cells. This is called as lateral intercellular spaces. And one of the important point of this is there are the luminal side of the cells have brush border due to microvilli. This is the proximal convoluted tubule. The proximal convoluted tubule, the next part which we get is the loop of Henle. Loop of Henle has an ascending limb and a descending limb. This ascending limb is very thin and a part of the ascending limb is thin. These two parts have a thin permeable cells. Thick ascending limb has thick cells with many organelles. So one of the findings which we have to learn in the loop of Henle are the two type of nephrons, cortical nephrons and juxtamedullary nephrons. Cortical nephrons, they're present in the cortex, where the glomeruli which are present in the cortex, they have a short loop of Henle. They're called as cortical nephrons. Juxtamedullary nephrons, they are located in the juxtamedullary head, then they have a long loop of Henle. This thick ascending loop of Henle, it extends up to the glomerulus. From there, part of the tube comes in contact with the afferent and efferent arteriole, predominantly with the afferent arteriole. And this part of the tube which is in contact mainly with the afferent arteriole, have specialized cell. This is called as macula densa. So one thing we have to remember is this macula densa cells with renin secreting cells within the renin secreting granular cells within the afferent arteriole, along with Lassie cells, they form the juxtaglomerular apparatus. Next comes the distal convoluted tubule. Distal convoluted tubule is about 5 mm long. Then it continues as the collecting duct. The collecting ducts are 20 mm long. It passes to the renal cortex, outer medulla, inner medulla, then empties into the pelvis of the kidney and the abscess of the medullary pyramids. This collecting duct, there are two types of cells, principal cells and intercalated cells. Principal cells also called as P cells, intercalated cells also called as I cells. These principal cells are tall, they have few organelles. Intercalated cells, there are many organelles, many mitochondria and cytoplasmic vesicles. So what are the importance of these principal cells? It causes sodium reabsorption, and vasopressin stimulated water reabsorption occurs. So they are important in sodium reabsorption and vasopressin stimulated water reabsorption. And intercalated cells, what are the importance of intercalated cells? It's useful in acid secretion and bicarbonate transport. This is about the nephron. Thank you.